Hello and welcome to Chat GCP, a medical science, clinical research and commercialization podcast, and I'm your host, Charles Beasley. On today's episode, we're lucky enough to have Dr. Sarah Diermeier. Sarah is the Chief Scientific Officer and founder of Amaroq Therapeutics, a venture-backed preclinical startup biotech company based right here in New Zealand. The world of biotechnology and drug development is incredibly competitive and highly complex. In this world, Sarah stands out as somewhat of a unicorn. In short order, she has completed a PhD followed by a post-doctoral research assignment at a prestigious institution in the US before starting Amaroq Therapeutics. On today's episode, you can expect to learn why our junk DNA might not be junk after all, the function of long non-coding RNA in many normal biological processes as well as disease progression, and finally, what Amaroq is doing to leverage the discoveries in long non-coding RNA and the development of the next generation of RNA therapeutics for oncology. So now, ladies and gentlemen, please sit back and welcome Dr. Sarah Diermeier. Welcome to the podcast, Sarah.
Thank you so much for having me.
Man, I am so looking forward to this conversation. I think I mentioned to you a couple times now, which is seriously embarrassing, how much I'm fangirling over the concept of long non-coding RNA, which is obviously the mainstay of what Amaroq is. We'll get to that in due course, of course, because we've got plenty of things to cover. But why don't you start us off with a bit of a background about yourself, how you got into genetics research, and how you started in Germany, went to New York, and came to little old New Zealand to start your company and many other things, of course.
Yeah, sure, happy to. And of course, it's perhaps a bit of an unusual journey. I probably didn't expect to end up in New Zealand, but here I am, and very happily so. Yeah. It all started probably about 25 years ago when I was a teenager and had a family member by my Aunt Elizabeth had cancer, bowel cancer, and succumbed to the disease sadly a few years later. And then my grandmother also had cancer and also died shortly after, and all of that happened within the span of a few years during my teenage years, and was really a significant event in my life in many ways, as you can imagine. And I remember, as a teenager, I felt really helpless because I saw my relatives were suffering from chemotherapy and radiation, the typical standard cancer treatment, especially we're talking, 25 years ago where there was very little else available. So terrible side effects, nausea, hair loss, the whole shebang. And then they died. So it didn't even help them, right? It didn't even improve patient survival. So I was thinking, very naively so at the time, of course, well, if those doctors can't help, well, then I need to do something. So I went to the library because back then there was no internet. So I went to the library and tried to find out what can I do to become a medical researcher to understand how drugs are made and how can I make drugs for cancer. And I learned back then in the school library in Germany that, well, first of all, I should probably complete school, but then I can go on and study biochemistry and get a PhD. So that was really important, become a doctor, and then you can go and make drugs. Or so I thought. Well, I guess it wasn't entirely wrong either. So I did that, right? Yeah. Finished school, studied biochemistry, did a PhD, and then I didn't immediately make drugs. Because by then, of course, many years have passed, and I had realized that I quite enjoy academic research and understanding more about molecular biology and how cells work and how organisms work, and wanted to learn more of that first. So I decided to move to the United States. That was another serendipitous event in my life. I went to the States for a conference, and then one of the conference organizers liked my talk so much they offered me a job. And I said yes, because why would you not? So moved to New York.
When someone offers you a job in the States, you...
Right. And in New York, like, oh my God. I was in my early 20s. It was fun times. So I moved over there, and I happened to work in the lab of Professor David Spector at Cold Spring Harbour Laboratory, and he's of course a world leader in the space that I'm now working in. Was very, very important. It still is very important for my pathway and for Amaroq as well. And that's where I went more into the medical research side of things, learning a lot about cancer biology, learning a lot about non-coding RNAs, which we'll talk a bit more, I think, in a little bit, and also drug development. So it was a collaboration with Ionis Pharmaceuticals, which is a drug development company, and together I got, I think, the best of both worlds during my postdoc. I also met my husband there. So he worked for the same institute, arrived at the same time. We started off as flatmates, then became a bit more than just flatmates, and then fast forward now we've been together for 10 years, have 2 little kids, and are married, and decided that after 5 years in the United States, it's time to, settle down, buy a house, have kids and stuff like that. And New Zealand seemed to be the perfect country to do just that. So here we are now in Dunedin, living the life.
Absolutely, and New Zealand is better off for it, I would say. Yeah. So you started off in proper blue skies research, molecular biology, figuring out how things work, the fundamental science, the biology, and now you've moved into more of a therapeutic biotechnology space. Was that a serendipitous transformation connected with Cold Spring Harbour, or was it always the intention to eventually, once you've cracked the code of this next big thing, moving into the biotech space?
I think it was always the intention that I want to have impact, right? I want to have a real world impact. I want to see the fruits of my labor at the end of the day. And of course, for some people that is rewriting the textbook and understanding molecular biology in the very detail. And I've done that as well, and I find it very exciting. But I always wondered in the fundamental research, how am I adding value to the world? Can I make a big splash? Is there anything that my research can contribute? And this applied angle was a bit missing. So I was quite happy that with David I could go into the applied world and really focus on how do we not only understand molecular biology but leverage that to change the world, to make new drugs.
Yeah, that's amazing. So why don't we talk about your field of particular interest, which, again, massive fan of. Very cool. But that is long non-coding RNA. That is a topic that we could get lost in forever. But why don't we, for the uninitiated and listening in, maybe give a high-level overview of maybe DNA, RNA, and epigenetics, and kind of the minimum set of requirements that we need to know to kind of facilitate a discussion around what Amaroq is looking into.
Yeah, for sure. So for most people, when you think about genes, genomes, inherited information in the cell, you think about DNA, that's correct. And then you probably go back to your high school biology knowledge and thinking about the central dogma of molecular biology, as we like to call it. So that means a gene, so a piece of DNA, is transcribed into a messenger RNA molecule, which is then translated into a protein. And then those proteins have functions, right? Enzymes, for example, is a good example from the school textbooks. And that's probably where most people stop thinking. And by most people, I also mean big pharma companies, for example. But that's not actually true. This is far from the complete picture. In reality, only about 1 to 2% of our genes, of our DNA, makes proteins. So that means there's 98 to 99% of DNA that does not make a protein. So what does it do? Anything?
I mean, that just, those two sentences just blow my mind. It's almost like we've got it around the wrong way. We're looking at the 1% and we're talking about, which is the genes that code for a protein, and we're talking about that 99% of the time, and foregoing the other 98% of what is actually in there. And that's been coined the dark matter of the genome, right?
Yes, exactly.
So it's there, don't quite know until recently what a lot of it does.
Exactly.
It's absolutely amazing. So, but within the non-coding parts of the DNA, so the junk DNA, what do we have? I mean, obviously it's there for a reason.
You would think, although, to be fair, that's hotly debated in the field. There are still people who say, some people argue if it's there, it needs to have function. Other people argue just because it's there, it doesn't need to have function. There could be evolutionarily remnants of viruses and, that kind of stuff. So it is actually hotly debated. However, I'm personally more in the field of if it's there, it may have some function in a certain circumstance, right? Maybe not in every cell, maybe not in every tissue. We might have to study it, but I think under certain circumstances, all of that will have function. So what's there in the DNA that is not protein coding, those 99 to 98%? Some of it is DNA that is not getting transcribed. And many of these elements have epigenetic function. So that means they drive in one way or another the expression of, for example, a protein coding gene, regulate when it's switched on and when it's switched off, for example. So that makes sure that, for example, a liver cell knows that it's a liver cell versus a heart cell knows it's a heart cell, right? So just switching on the right proteins, the right genes at the right time in the right place. But there's also a very, very large percentage of the genome, so about, numbers vary, but about 75%, I think is fair to say, that is transcribed. So makes RNA, but this RNA is never translated into a protein. And this is where we're talking about non-coding RNAs.
Right. And within that non-coding RNA, there are a bunch of different subtypes. I think everyone that's done maybe high school or university biology would have heard of tRNA.
Yep, exactly. That's a good example.
rRNA, ribosomal. RNA,
Exactly.
Yeah, not looking at my notes, people. All right.
No, those are great examples indeed for non-coding RNAs that most people would have heard about, but don't really think about them as much as what do they do and why are they there. But yes, of course, they are essential for mRNA to be translated into proteins, so clearly they have a function, right? I think that's undisputed. And then there is a lot of other types of non-coding RNAs. MicroRNAs, so the short ones, have been hanging around for a while, and I think most people have now accepted that they are a thing and might have some functions in certain circumstances. And then, of course, there's my favorite topic in the world, called long non-coding RNAs, which is really just, it's longer than 200 nucleotides, doesn't make a protein, that's it.
Yeah.
So very ambiguous definition, leads to a very heterogeneous group.
Nice. Is there a likelihood, do you think, that eventually they'll be so well studied that they'll be branched out into different groups? Because, like you say,
so.
it's very broad.
I hope so, because actually the breadth sometimes is confusing, because you're talking about tens of thousands. Some people say 100,000 different molecules, right?
Yeah.
And I think subcategorizing would be really helpful. But of course, how, right? And I think that's one of the biggest questions that the field will address in the next decade of non-coding RNA research.
So out of all those non-coding RNAs, is the long non-coding, are they the most recently discovered category?
Yeah, I'd say so.
Yeah.
They're also the largest and most diverse.
So if they're largest and most diverse, you would think they'd be discovered first?
Yeah.
Or is it very specific techniques that are required to elucidate?
That's exactly right. So one reason why we didn't know that they exist and make up the majority of the genome, in fact, is that we could only find them thanks to new technologies. That includes high-throughput sequencing, for example.
Yeah.
So for the past 10, 15 years, we've been more aware of what's out there, but before that, it wasn't even possible to find them.
Amazing. So surely that would be like stumbling on a gold mine for someone in the field like yourself. You know, this is something that we haven't known until recently. Technology has made us aware that there is a completely new avenue to green grass to be,
Absolutely.
investigated.
Absolutely. And that's how I think about it too, that from a drug development perspective, right, all of these are clinically unexplored, uncharted territory. So for me, this is just amazing potential to study, to learn, to find out which of those could be, the answer to cancer, the answer to any type of disease.
You're like a genetic Magellan.
Yes.
Finding
nice
new
comparison.
lands. Okay, so we've got, the genome, 98% of it does not encode for a protein. I think we can forget about the 2% because we all know that from high school, if not university. Of that 98%, there are different categories of non-coding RNA, the most recent of which is this long non-coding RNA, fairly broad category, greater than 200 nucleotides. What do they do?
Yeah. I mean, what do they do?
Do we know?
We know, actually know, as in we have scientifically shown for maybe 1% of those long non-coding RNAs, right? So really, if there is 100,000, let's say we know about the function of 1,000, so about 1%. Not very many. And they do so many different things. Like, we've talked about tRNAs and ribosomal RNAs. They all do the same thing. Well, maybe they don't. Maybe I'm oversimplifying, right? But let's assume for now they all do the same thing. Protein synthesis. MicroRNAs also more or less do the same thing. Long non-coding RNAs or lncRNAs, they all do something different. There have been some that have been described in epigenetics, so bind to transcription factors, chromatin remodelers, splicing, or also in the cytoplasm. They can be involved in metabolism, they can be involved in RNA stability, in translation. There's really, show me a function where they've not been implied, right? That's really the challenge, perhaps. They do anything, and that's what makes it interesting. And you have to study each one that you find individually and with detailed molecular biology to find out what do they do. And this is where the background in fundamental biology comes in handy.
And it wouldn't be just, okay, you've identified this lncRNA and you know what it does, but it would be tissue-dependent, organ-dependent.
Yes.
Time-dependent.
Yep, development-dependent, right? Might play a role in early embryonic development. Then that happens actually quite often, and they're switched off in the adult tissue. But in cancer, they might be switched back on, and oh no, they're detrimental to the cell now.
Yeah. So how these people that argue that they don't do anything, they're not long for this world, surely.
Oh, you'd be surprised. There is some bioinformaticians who've been in the field for decades, and I think maybe just don't want to change their opinion. I don't know. Perhaps. No, I mean, they have their arguments, and I respect that, and I accept that. I think we've shown for many lncRNAs over and over again that they have important function in the cell with biology.
Yeah.
So computational arguments against it, then in my mind, are just slightly less convincing. But that's my personal opinion, and I accept others.
Yeah. So this opens up a whole potential new avenue of not just original research, but also then applications.
Absolutely.
Right. So we can talk about Amaroq in a minute, but again, absolutely fascinating, amazing. So are there any well-known, well-characterized examples of lncRNAs, really just to drive the point home?
Yes. One good example, I think, is Exist. So Exist is a long non-coding RNA that's transcribed from an X chromosome in female mammals, so organisms with two X chromosomes. And there's a problem if you've got two X chromosomes that really you would have all genes from both X chromosomes expressed at all times. So what needs to happen is that the cell needs to shut down one of them. So dosage compensation, right? And to achieve that, so that only one X chromosome is active, Exist is expressed. It's sitting on the X chromosome, and it silences one of the X chromosomes through an epigenetic mechanism. And now it's switched off. It's the bar body hanging out there in the cell, not doing much. So absolutely essential in embryonic development, absolutely essential for any female mammal with two X chromosomes. So I think that is one of the great examples where we have shown beyond any reasonable doubt that there is a function for this dark matter gene.
In my mind, in my 5-year-old with a crayon drawing on a wall idea of this, you've got this very long RNA molecule, long non-coding RNA molecule, and does that wrap itself up into different shapes and interact with the different nucleotides to different parts of it? Might be protein in origin, it might be DNA in origin. So it's like a scaffold / interacts, turn things off, turn things on. It can be, yes. So you're quite right, it folds. And I think that's true for all RNAs, also your messenger RNAs. They will fold in all sorts of ways because things don't actually exist in one dimension, right? Wait, hang on. What?
Every biological molecule will have a 2D and a 3D structure, like a protein, right? Isn't just an amino acid chain that's hanging out there. So same for RNA, folds up and then can be a scaffold, for example, for binding of transcription factors or epigenetic regulators. But that might be oversimplifying some of its functions. So again, it comes down to the individual lncRNA. Some might act as scaffolds, other might act as ribozymes, right? So we have an enzymatic activity within the RNA, like your spliceosome is a ribozyme, for example, or your ribosome is too.
Fantastic. So why don't we pivot to your research that you did at Cold Spring Harbour, because that was when you that was your more or less first foray into the cancer side of things.
That's right.
Yeah, and the potential application of the lncRNAs to the treatment of cancer, which, correct me if I'm wrong, but that is a completely new, previously untested before then
Absolutely.
treatment modality. Yeah.
Absolutely. So, and perhaps it's worth noticing that up front that Amaroq is one of the first companies in the space, and 3 years ago when I raised the seed round, there was no competitor, right? There was no company in the world working on long non-coding RNAs in any indication. This has changed now. Can talk about that later. But yes, we definitely are pioneers in the field, and that's on a global level. And that is really because I was lucky enough to work with David Spector at Cold Spring Harbour Laboratory, who is one of the world's absolute pioneers, who is one of the first researchers in the world who identified long non-coding RNAs and their function in development as well as in cancer.
Wow, that's so amazing. And you did breast cancer research,
Correct.
Is that right? Yeah, Cold Spring Harbour. And
Yeah.
what were you showing in that research? Well, I was going to say trying to show, but
Yes.
you did show.
We did show some things, yes. So when I joined David's lab, he was working on one particular long non-coding RNA called MALAT1, and that has been his baby for much of his career, right? But what we hadn't shown was a function for it, and that was quite interesting too. So he even made knockout mice that didn't have the gene, and they were perfectly fine. So that, this is where we're coming back to the controversy. Do long non-coding RNAs actually do something? At this stage, we were like, oh no, we made this whole knockout mouse. It does nothing. How can it not do anything? This is crazy. So when I joined the lab, there was a project with another postdoc, Gayatri Arun, and she crossed these knockout mice into a breast cancer model. And that's when we started to see the phenotype, which is why I am convinced that most junk DNA or junk RNA isn't actually junk, but has a function if you test it in the right condition.
Sure.
We just had to find the right condition, which was breast cancer. So we saw that in these breast cancer mice where we didn't have the long non-coding RNA, the tumors were much smaller, they were very differentiated, and the mice had no metastasis to the lungs. So that was really, really striking, a really amazing phenotype. And we then went on to develop a drug, an Antisense oligonucleotide drug, to target this lncRNA MALAT1 in collaboration with Ionis Pharmaceuticals, and tested that in those tumor-bearing mice as well. And we realized that we can drug the lncRNA with the antisense in a systemic manner, and we can then reduce tumor growth, reduce metastasis, and induce tumor differentiation. And that was the first study that I'm aware of that has shown very conclusively that lncRNAs play a role in tumor progression.
Amazing. So essentially, if you didn't have that lncRNA, the tumor did develop, but not quite as aggressively, or not quite as...
Correct, yeah. So
and then you add a antisense oligonucleotide, which knocks down any activity with that lncRNA,
That's
and
right.
then it goes back to the baseline, essentially. Fantastic. And since then, that was the pivotal research at Cold Spring Harbour.
Yep.
And then you've brought a lot of this research, or built on this research, in your role at Otago University.
Yes, that's right.
Yep. And of course, we'll get to that eventually, I promise. There's Amaroq. But so maybe briefly, the continuation of your research at Otago, like what's been happening there recently?
Yeah. So when I joined David's lab, we were researching breast cancer, but different types of breast cancer, right? And we were really focused on estrogen receptor positive types of breast cancer because they're the majority of all breast cancers. What I lacked at that stage was a bit more commercial insight, because I learned towards the end of my postdoc that actually new drugs for this particular very common type of breast cancer are hard to market because there's already really excellent drugs out there. And what I really should do is look into the types of breast cancer or other types of cancer that don't have a lot of good treatments available.
Right.
So when I came to New Zealand and started my own lab at the University of Otago, I set out with a bit more of a commercially trained mindset perhaps, and I'm like, okay, where are the gaps? Which types of cancer need new treatments most urgently? And let's focus on that. And that's what we did, and started with triple negative breast cancer. Again, breast cancer, so bringing all my expertise to the table, but now looking at the one type that's super aggressive and doesn't really have any good treatment opportunities yet.
Yeah, so that commercial mindset really set the stage for you to start Amaroq.
Absolutely.
Yeah, because you're going from fundamental research, and then with a commercial mindset,
Yes.
the logical progression is to let's spin this out into a commercial entity that will fund it to eventually be administered to patients. What was that journey like? Was it serendipity that opened up the avenue to start Amaroq? And how did that start?
Yeah, it was to some degree. I have to say, startup curious. And that's, again, from my time in the States. Of course, the closeness to New York City, there's a lot going on,
Yep.
lots of startups. Many of my friends back at Cold Spring Harbour started their own companies, which I really admired, but wasn't quite sure if it was meant for me. But I at least had some examples, right? They did it, then surely, maybe I can do it, maybe. Maybe not. And another interesting moment in that career was we invited Jennifer Doudna to Cold Spring Harbour. And she's, of course, the CRISPR lady, if you like. And she made such a huge impression on me because she talked about how she's a professor and a startup founder. And that was the one where I was like, oh my God, I want to be like her. Absolutely fangirling over her, and decided, okay, let's try, right? Let's just see what happens. So when I started in New Zealand, in Otago, within the first 2 months of arriving, I went to our TTO and said, hello, I have this great idea. How about we're targeting long non-coding RNAs and triple negative breast cancer with ASOs? They looked at me, you can
Speaking
imagine.
another language?
Here's this crazy woman that just arrived from the States with this German accent. She's talking about things we've never heard before. Oh no, what do we do with her? So it took a little bit to get that going, let's put it that way.
Yeah.
Get some traction and convince them that, yes, I have the expertise to do that, and there is a market for it, and this is totally possible. But we got there eventually. Got some traction about 2 years in, and ever since it was relatively smooth sailing, actually.
Well, that's fairly unusual in the startup space.
I know.
So, well, kudos. So, founded in 2021. So the tech transfer office, you managed to really twist their arm into seeing.
They came around eventually, and once they did, they were super supportive ever since. It just took this initial activation energy to convince them that this is something worthwhile their time and money.
And there was also some investment involved as well with a
That's
consortium of like startup activators and that kind of thing that helped. Obviously, science is not cheap to do.
No, not
research,
at all.
clinical research, not cheap. Preclinical research, not cheap. So
Drug development is very, very expensive, very capital intense upfront, and it's scary for some investors, I'm sure, because the lead times are so long until there is an exit, and there might not be one because it's so high risk, right? So
we've all heard of those. Sorry, we've all heard of those graphs where it's like time versus approval, and it's like potential 10,000 molecules for 10 approved drugs at the other end in a 20-year time frame.
Yeah, and how one drug to the market costs $1 billion, and
Is that all?
I think it depends on how you crunch the numbers, but on average, probably pretty close. Yes, but we were lucky in the sense that I participated in a pitch competition at the uni, translational research grant, where you can just pitch your ideas, and you've got an audience of some investors. And luckily for me, a representative of Brennan Capital, Duncan McIntosh, was in the audience, and he got interested. And that was really what turned the tide for me, because once he was interested, the TTO was interested, and then everyone was interested. And it still took a long time, to go through the due diligence, and it was a very intense period to get to the point. But we eventually got that seed funding across, led by Brennan Capital, and with support from Innovation Booster, QKids Ventures, and Otago Innovation. And there was a very sizable seed round, especially for New Zealand standards, very sizable. So we are very lucky. And the science has been going well since. Yeah.
Not looking back. Was it a case of, you mentioned activation energy. I love that phrase. That's so good. It's almost like, but you also just need to get started. You need to get the ball rolling.
Absolutely.
And it kind of sounds as though as soon as one person put their hand up and said, yeah, I believe in this, everyone else, a bit like popcorn, goes, oh yeah, totally. Absolutely.
Yep, that's exactly right. You need that one supporter, that one investor, that one person that believes in you. And all of a sudden, you're a lot more attractive.
It's a vote of confidence, essentially, isn't it?
Absolutely.
Yeah, as if like all your previous work wouldn't be enough of a vote of confidence. But when you're dealing with commercial aspects, there's not always a one-to-one parallel, right? I mean, things don't translate well. There's almost like a screen that you have to squeeze yourself through, and only part of it gets out the other side.
Absolutely.
It's a different version.
Yeah.
So for you, going from a fundamental scientist into a commercial operator and chief science officer, what was that like in terms of your approach to work? And did you have to relinquish things that you otherwise would not have wanted to?
It is certainly very different, right? In your academic job, in my academic job, I'm all about curiosity, understanding biology. It's not about selling anything. I don't have anything to sell. If I go to a conference, I tell exactly what I did and how it went. And if it didn't go well, I tell that too. Now, in a commercial setting, I have learned, first of all, it's a different language. There's also different measures of success, right? And it's all about telling a story, storytelling, selling, things that might not come second nature to an awkward academic, perhaps.
You're not telling a story when you're doing academia, are you? You are telling it like it is.
Exactly, facts.
Yeah.
Right, here's what
That's
I did, didn't work. Now you know, it's not worth doing.
Definitely. And the language of science around we have evidence that would suggest does not translate well in a commercial sense. And for the people that don't understand what that language represents, it goes, they say, what are you saying?
You are so right, and I think this is my biggest struggle to this day, is when I talk to investors, I'm so deeply ingrained in the academic mindset that I'm very hesitant to say, and this blows out all of the other drugs out of the water because this is amazing and we're amazing and this is the best thing ever, right? I just don't use that language.
You're not allowed to. That's so interesting. And you've got different stakeholders as well.
Absolutely.
So you've gone from your, I suppose in academia, I've never been an academic, but you've got your department, make sure your funding is all good, satisfying those responsibilities, but commercially, you've got your investors.
Yeah.
Yeah. Obviously, you've built a team around, to actually do the science. Have you ever felt that you had to let go of the control a little to make this happen? I mean, you've got to go from being immensely curious and answering questions that are just for you to answering questions from other people.
Definitely. It is a little bit letting go of control and a little bit of taking the commercial overarching goals, take the front seat. And of course, you've got investors. They have questions. You need to answer them. They have ideas. You need to have at least a response of why they are not good or why they're good and then do them. So there is definitely, it feels a bit more controlled. Of course, as an academic, you imagine this free-spirited professor that just does whatever they want all day long. It's not quite like that either, because you're somewhat restricted in what you do by grant funding, right? So there are some stakeholders there. There can be government agencies, for example, and you still have to do the work that you promised to do. And maybe you can't do exactly what you want to do, but you have to make it fit the funding call. But it is a lot more restricted in some sense what you're doing in a commercial setting, of course.
Yeah. And there's kind of really concrete set timelines as well.
Very.
And either dictated by how much money you have or expectations from investors and other stakeholders, right? As opposed to we'll get to it when we get to it based on what we're doing in the science.
And I think that's been a challenge for some of my team members as well, switching from that academic mindset where you're like, oh, this didn't quite work, but maybe we try it this way and another way. And there's this other essay that I read about, and you just keep trying and keep doing and digging into that hole. And in a commercial setting, it's more, this didn't work. We are not going to do it again because we cannot, we don't have the time, we don't have the resources, this is parked or this is dead, and we move on to the next thing. So, it's a big difference in how you work.
So let's talk about the research that you are doing right now at Amaroq. So Amaroq, it's preclinical stage, so you're in the process of identifying potential lncRNA targets in cancer. And once you've identified those, you would then go through a series of additional steps to prove that they're good candidates, validate the candidates, and then eventually preclinical animal trials. And then once you've done that, we're into clinical trial.
Clinic, exactly.
Yeah. So why don't we talk about the current status? Where are we at right now?
So we are in the preclinic. We have not started any human trials yet, and we are a couple of years away from that still. But we have built what I believe to be the world's best target ID platform for long non-coding RNAs in oncology, but can be used for any indication. And we have validated 2 targets, one in breast, one in bowel cancer. So that means we have done a lot of experiments in vitro in cell culture, but also in mouse models that show that these new targets are driving cancer growth. And we've also developed drugs, so these Antisense drugs for these 2 lead programs, have tested them again extensively in cell culture and now also in animals. So we are basically coming to a big inflection point for us now, where we have shown our pipeline can identify novel lncRNA targets, and we can make drugs to target them with a tumor response in vivo. So that's really fantastic, because that means we're ahead of what everyone else is doing in the field at the moment, and are probably ready to partner soon.
So it's almost vertically integrated. You've got the identification pipeline, the validation, and then
100%.
moving into that. And that, like you say, increases the pace of what you can do. You don't have any other, even more stakeholders trying to throw their
No, that's right. We do everything in-house.
desires in the mix.
And we could, in theory, take everything up until the end of phase 1 clinical trials. So we are resourced to do that and have a pipeline to push assets along.
And you mentioned the commercial aspect around the breast cancer that you've... So the triple negative, is the same applies for the... colorectal cancer target that you've identified?
Yeah, that's right. So bowel cancer also sounds like, there should be a lot of good treatments around, and there are some, but there aren't enough. It's still a big unmet clinical need. And that's why we decided to go into the bowel cancer space. It's also a really big medical problem in New Zealand in particular. I wasn't aware of that before I moved here, but apparently highest rates in the world, or some of the highest rates in the world, especially in women. Yeah.
Really?
Yeah, right?
Yeah. We've got melanoma and that.
I know. And
go to New Zealand, right?
Bowel cancer. I don't know. So yes, so we decide based on high unmet clinical need, which is why we prioritize breast, bowel, and the next one in the pipeline is for glioblastoma, which is a very aggressive type of brain cancer.
Yeah, huge unmet need with that one, right? I mean,
absolutely.
That is devastating very quickly as well.
It is, yeah. You have, within a few months, basically, the patients are wasting away. The challenge with glioblastoma is a bit on the commercial side, that it is not as big of an indication, so the market's smaller, right? And therefore, the potential economic benefit for a partner might be smaller. But you're quite right. It's such a huge unmet clinical need that we can't not do it.
Yeah. Okay, so... those are the 3 main candidates in various different stages. The most advanced one is the breast cancer target. Yep. And so we're thinking 2 years away from potentially clinical trial.
I think that's realistic.
Oh, that's light speed fast,
Yeah.
right?
And you know why? Because this new type of drug that we're developing, these Antisense oligonucleotides, they are much faster and cheaper to develop than your protein drugging small molecules, for example.
Because they're highly predictable and selective, right? A small molecule kind of goes everywhere and interacts and bounces around.
Yep, exactly. Yeah. And a small molecule, you need to do years of chemistry to get from a lead to a development candidate and a clinical compound. With Antisense oligos, you can optimize much faster. You have a lot less to play with, and it's a lot more specific, a lot clearer how to design it and what to do to move to the clinic. So that's how we are so fast, and that's how we can operate on relatively low capital.
Yeah. So these antisense oligonucleotides, actually going back to the lncRNA, these remain in the nucleus? They don't go into the cytosol?
They can.
They can, okay. Yep. But the ASOs can go into both.
They can go into both. And this is why we're using antisense oligos, because they can work in the nucleus and in the cytoplasm. And the lncRNA may be in either or both compartments. Right. Again, depends on the target.
Whereas some other alternatives might be the siRNAs, but they cannot get through to the nucleus. Is that right?
That's right. They can only act, again, this is another heavily debated topic in science, but generally accepted view is that they can only act in the cytoplasm. Yep. So if you've got a nuclear RNA, you have less luck targeting it with an siRNA likely than with an ASO.
Right. And the ASO, what is their mechanism of action, So they bind onto the lncRNA based on a certain sequence that they have. and then that gets chopped up.
Yeah, that's
And,
right. So the reason why we can make them so fast and easy and cheap is because we know the sequence of the RNA, right? So we make a sequence complementary antisense, and that binds to it. There's different types of mechanisms of what happens next. For our approach, we use what we call gapmers. So you're quite right. That triggers an endogenous mechanism that then degrades the RNA. So RNA is gone. Happy days. Cancer cell hopefully dies.
Yep.
And there's other types of antisense oligonucleotides that can do other things, like impact splicing, for example, or act as a steric block. But we're not currently using those.
Oh, incredible. And you can target ASOs to different tissues, right? I know you can do that with siRNAs. You add a little whatever it is, a little target onto it, and it goes to the liver or it goes to whatever. And you can do the same with ASOs.
Yeah, you can do the same. You can chemically
So
conjugate
again,
them.
additionally, very targeted. Not just based on the sequence, but even the tissue it gets to.
Yeah, absolutely. And it depends on where you want them. So, for example, if you want them in the CNS, best to inject them into the CNS. They will not cross the blood-brain barrier, for example. And
So lumbar puncture kind of thing, or directly into...
intrathecal works.
Oh, okay.
So there is a number of antisense drugs that are on the market that are delivered intrathecally. You could also, if you wanted to treat brain cancer, you can go in at the surgery site, for example. And liver, yes, for example, is a very good example how we can get antisense oligos with a little chemical modification that then very specifically go to the liver only, like an siRNA.
Yeah. Oh, that's amazing. All right, so what would a phase 1 trial look like for these things? I can't imagine there'd be anything beyond the kind of run-of-the-mill phase 1 study. You've well identified, you validated the target, you'll have preclinical information to talk about its safety as well, and then you are just going to essentially ramp up the dosing to see if it's safe in people.
And we know from the other antisense drugs that are already approved that the safety profiles are really, really good. So in some ways, we are not too worried about safety or toxicity at all. And we have a good feeling for how to dose those patients. But yes, we would imagine that, say, if we start with triple-negative breast cancer, we can pre-screen which patients will be expressing the target, which based on our analysis is about 70-80% of all patients, but still good to pre-screen to make sure we only treat the patients that actually have the target and will benefit. And treat those. We could also think about a basket trial, is what one of our clinician advisors mentioned, where because this target, while we've identified it for breast, also plays a role in gastric cancer, bladder cancer, thyroid cancer. So it could be a way of enrolling more patients quicker and providing more solutions for more patients with a mixed basket trial.
Oh, amazing. Can we talk about how targeted it is again? Because I'm absolutely fascinated by this. Because this, like a bit like what you mentioned, the old school approach to science is build a small molecule that ideally targets some protein that we know has an effect in a particular disease, and it's sufficiently targeted to that protein and not many off-target effects that would compromise its safety. Whereas a bit like what you kind of alluded to. I mean, you're not worried about the toxicity of these ASOs because they are so targeted. They go straight to the tissue of interest. They go straight to the lncRNA of interest.
Yep.
Presumably no other off-target effects because it is that unless it has that sequence.
Yep.
What's the likelihood of something else having the same sequence as the lncRNA you're targeting?
Great question. So the way we design our ASOs, it's very unlikely that there will be an identical sequence. This is how we screen our targets. This is how we screen our ASOs. We make sure everything's as specific as we can possibly get it from day one. But it is also fair to say that there is a certain level of promiscuity in binding. So even if you don't have a perfect sequence match, if there's one or two bases different, your ASO might still bind to that, right? So that would then be some sort of off-target toxicity. That can happen. We do try our best to avoid that from the get-go, and we do a number of experimental assays to avoid that. But this is essentially the only real concern for safety or toxicity when we go into, say, rodents and non-human primates later, is that there could be expression of a target that it looks kind of like our target but isn't quite, and we get some off-target toxicity. I don't think the chance for that to happen is high, but we do have to acknowledge that there is a chance. Yeah,
and I imagine that the chance of that that happening would decrease the greater number of target bases on the lncRNA. You know, like how likely is it that you're going to have that exact sequence? I mean, it's like the Shakespearean, so the chimpanzees writing Shakespeare on a
Yes.
on a typewriter. Eventually, it's going to happen, but
Yep.
it's very, very unlikely. No, it's amazing. So maybe we can take a step back and go. So we've got those three initial targets that you're going after. But I can imagine if there are thousands upon thousands upon thousands of lncRNAs out there that are potentially implicated in not just cancers but other therapeutic areas, you're not just going to put all your eggs in three baskets, shall we say?
No.
Yeah. So you're doing additional screening to identify other lncRNAs, right?
Yes.
Yeah.
So we've developed this discovery engine, this pipeline, right? And what my hope is that we just keep cranking it, right? Run it through all types of cancer and just see what we get. See which clinically actionable lncRNAs we can identify for all types of cancer, but beyond cancer too, right? There are some diseases out there that really desperately need new targets, like in the neuro department, Alzheimer's, for example, where there hasn't been new targets in decades, right? And we're still trying to solve old problems, but haven't been able to solve them for so long that perhaps it's time for looking somewhere else, looking new. And this, I think, is where we can come in and just say, hey, let's run our pipeline on Alzheimer's data and see what we come up with and see if we can make a drug for that. And that can be repurposed for, any indication, really, in theory.
Incredible. And that the data that you would need, that's genetic data from patients with a defined condition.
Yep.
And then you essentially just run your,
Yes. So RNA sequencing is what we would be looking for. So as long as there's RNA-seq data from patients, we can run our pipeline on it.
Yeah. And you're collaborating on this with, is it GeneCrypt?
Yes, yep, exactly.
I always get that wrong. Sorry, Chris. I don't know why. Always.
I'm sure he's fine. Yes, so this is a very exciting collaboration between Chris's team and our team, where they use their encryption technology to encrypt our pipeline end to end. And I think, as far as we're aware, we might be the only biotech in the world currently having an end-to-end encrypted target discovery pipeline. And that's good in two ways. In one way, we have patient data, right, from New Zealand patients, and we want to make sure that that's absolutely 100% safe and secured, and the GeneCrypt technology can ensure just that. On the other hand, we've got a commercial advantage now because we can go to, say, a big pharma company, ask them if they want us to look into their data with our pipeline and identify lncRNAs, and we never have to disclose what we do or how we do it because it's all encrypted, right?
Yes.
So it really enables us in our partnering efforts, but it also ensures that all patient data remains safe.
That's absolutely remarkable. So another world first. You're really just
I
doing a
think
lot
so.
of world firsts over here, Sarah.
I like to be pioneering.
That's fantastic. Honestly, this has been an absolutely riveting conversation. Like I said many times, I am absolutely thoroughly impressed by what you're doing down there in Otago, and I wish you nothing but the best. If people want to find out more about what you're doing, Amaroq, yourself, where can they go?
You can visit our webpage at amaroqtx.com. I've also got a LinkedIn profile for myself as well as for Amaroq, and slightly less active Twitter or X now channel.
I don't know about that name, but yep.
I don't know.
Awesome. Well, thank you so much for coming on Chat GCP. I really appreciate it. All the best, and we'll be keeping an eye for sure to see what happens with Amaroq and yourself. Thank you.
Thank you so much.